Symposium. Welcome to Leuven. And for those who wouldn't know, oops, wait. So it should work. It's not working. Okay. So for those of you who wouldn't know, so Leuven is hosting the Catholic University of Leuven. And next year, so in 2025, we will be celebrating the 600th anniversary of the university. So this means it's quite old already, as you may have seen from some buildings that you saw yesterday during the walk. But let's come back actually to lobular breast cancer. I think that what is reassuring is that now, over the years, we kind of see a very continuous increase actually in research on that type of cancer. And this is, for example, a search I did 2 days ago on PubMed for searching for lobular breast cancer and lobular breast carcinoma. And you see that really the publications continue to increase, that the number of groups involved also continue to increase. We still need to do better, but that's already a very good sign. I think what we also can say is that finally also we get some increased attention by larger conferences, by other organizations, and I think in the past years, it's very recent, we did have some dedicated sessions on lobular breast cancer in these large conferences, for example, or that we will have. So there is finally also a recognition from the larger community that we need some dedicated attention. So actually, this year I just wanted also to give you, before I go into the practical information, I just wanted also to give you some information about the symposium. So we decided to have a little change in some of the sessions that we have. So we obviously will have fantastic keynote speakers. You will discover them along these 2 days. But then also we decided this year to introduce breakout sessions. And the breakout sessions we will have, you will see this afternoon, 1 block of breakout sessions, and tomorrow we will have also another block. So this is because we believe that while doing this effort to have the only lobular breast cancer symposium together, when we had this opportunity of having all these experts and people interested in doing better for lobular breast cancer, it was also good maybe to take the time to focus on specific questions and specific challenges. And this is why we were making some very specific organizing panels for each of the challenges that we were identifying. And for example, today we will have that on pathological diagnosis, we will have it on imaging, and we will have it on dormancy, 3 topics which we know are very specific and needed for lobular breast cancer. And you need to understand that all these breakout sessions have been prepared already very well in advance by all this organizing committee, which we made sure was always composed by a group of clinicians, scientists, and patient advocates. So it's a new effort, and we welcome everyone then attending these breakout sessions to be also very participative, you know, and we hope that this will generate new insights, new collaborations, and maybe also new works to be done within the field. Then also something that we will introduce this time is that we will have these short pitches, you know, from our poster presenters today, so that people could briefly hear also what all the different posters are about, and they could be triggered also to listen and to talk with these different poster presenters during the poster session this evening. And then finally, we will also have this speed networking event just before lunch, but we will tell you more together with Stephanie Walker later today about that. Don't stress. So now some final practical information. So the Wi-Fi, I think everyone has found. It's also on all the doors and so on. So again, for those of you who require some quiet room, you can go to conference room 6 here at the back of the auditorium. All sessions are recorded. Unless, and will be posted unless the speakers didn't give their consent to. And as yesterday, I would really ask everyone to enter the auditorium a few minutes before the session starts to make sure that we can start on time and that the speakers are not disturbed. Also, I will ask everyone to sign the attendance list every day. We need that every day, please. And then I would ask the speakers and the chairs to make sure to respect the timing of the session, because we have a very full agenda, and I think it's nice to make sure that we can keep the breaks so that people can talk further then and relax. And then finally, at 11, so just before starting the coffee break, we will have a group picture that will take place in the courtyard there. So please, before grabbing your cup of coffee and so on, go there. You will need to look up. We'll take a picture from the window so that we can try to have everyone on the picture. And so now, actually, it is, and also what important is, it's not the last day, but I absolutely also wanted to thank the scientific committee for the help in putting the program together. I wanted to participate, to thank the very active patient advocates for really also putting this symposium together with us, for all the meetings we had, for all the very nice interactions we had also in the development of the many sessions. Then a very, very big warm thank you to the local organizing committee, because without them, we could not have had this symposium. And then finally, also, I would like to thank the sponsors, which you see listed on this slide, but also on the banner. So also a very warm thank you. So now I actually would like to introduce the Dynami Foundation. So the Dynami Foundation actually will be supporting the lecture, which will take place in a minute. So the Dynami Foundation is led by Flora Mijianka. I'm not sure I'm pronouncing well. She's a very enthusiastic and passionate person who is dedicating her enthusiasm to try to gather money and attention to lobular breast cancer. And recently, they have developed what they call the Leigh Pate Memoral Lectureship. And this year, they have chosen Professor Patrick Derksen as the person who would benefit from this lectureship grant on the occasion of our annual ILC Symposium. And we are extremely proud of that. So Patrick actually obtained his PhD at the Department of Pathology at the Academic Medical Center Amsterdam in the Netherlands. And following his graduation in 2003, he moved to the Netherlands Cancer Institute to study the consequences of inactivation of cell-cell adhesion in breast cancer development and progression. And there he developed 2 mouse models for human invasive lobular carcinoma, biconditional and mammary-specific knockout of E-cadherin and P53. This is really a very important work which has set the basis for a lot of work on lobular breast cancer. In 2007, he joined the Medical Oncology Department as an independent researcher and head of the UMCU Mouse Cancer Clinic. And in 2010, his position was 10 years as associate professor at the Department of Pathology. Today's group is aiming at combining expertise on adhesion molecules, cell signaling, and clinically relevant mouse models to study the downstream effectors of the adherence junction complex that propel breast cancer development and progression. And we know, obviously, that he has a very strong focus on lobular breast cancer. He's also the person who started the European Lobular Breast Cancer Consortium, ELBCC, that Anne Salomon and I proudly are co-chairing. ELBCC aims at better understanding the biological cues that underpin ILC, identify and tackle clinical challenges to improve diagnosis, treatment, and prognosis of this disease. And importantly, since the start, ELBCC is also closely collaborating with many advocates to try to establish a strong EU-based ILC advocacy movement. And finally, Patrick is also a scientific advisory board member of the Lobular Breast Cancer Alliance. So it's therefore my great pleasure to invite Patrick to deliver his lecture here today. Thank you.
Okay, so good morning, everyone. We all know Leigh Pate and the magnificent work she's done for lobular breast cancer and, of course, her legacy. So I'm incredibly humbled and flattered that I was asked to do this presentation today. The second European ILC meeting, again in a church. I don't know about you guys, but I feel enlightened. I love to preach, so we're good to go, I think. So today what I want to do is tell you a little bit about lobular breast cancer, how we, or I think, functions from a biological point of view. I will limit myself to the work that is formally experimentally addressed. I will not go into much detail, but if I do, you can forget about them. All you have to do is memorize the concepts that I want to push across and which I think are important for clinical intervention. Okay, so this is the slide I always show at the beginning. It's also because Anne Salomon always tells me morphology is magic and is really important, and you should look at it, and I fully agree with that, but it's also really important to look at the biology. And this is something that was published by Chuck Peru back in the day, defining the molecular subtypes of lobular breast cancer. And why is this important? Because these are the only cues that actually can intervene in a clinical process, so either ER intervention or inhibition by chemotherapy, or recently the immune checkpoint inhibitors. But this is not the full story. These two independent things have to, I think, be combined to have an actual good clinical diagnosis and treatment, and for that I think this is the actual situation. So we have two histological subtypes of cancer, of which lobular is one, a very important one, but also lobular consists of all kinds of subtypes, being the luminal one, the HER2-driven one, and the basal-likes, and that should be taken into effect when designing a treatment. Okay, so these two rascals have defined lobular breast cancer during the war era. So they defined a subtype which they call lobular. I think in hindsight the name is completely erroneous because it has nothing to do with lobules per se, but anyway, they recognized the phenotype and they published this as lobular breast cancer. So this is a bit of a better picture that you all know as well. On the top you see ductal breast cancer, and on the bottom you see lobular breast cancer. And a distinct feature of the difference is that ductal tends to grow in nests, have pushing and invasive margins, whereas lobular have these very particular single file, back in the day called Indian files, type of invasion. And you see, clearly can see single cells. It's a bit difficult to see here if you're not a pathologist, but you can see single cells. However, if you put them into a gel in an experimental condition in the lab, it looks like this, whereas NST gives you an invasive growth pattern that is collective. That is collective, like this. ILC has these single file patterns in single cells. And if you fiddle about with the medium, in this case it's a collagen gel, and you do something with the polymerization temperature of the gel, you can actually induce these typical ILC invasive file patterns. So that's really distinctive for ILC. A bit of history, and I will go back to that in a minute as well. So the first observation that E-cadherin is not present in lobular breast cancer was done by the group of Walter Birchmeier in 1993. You can see that here. This is, oh, I'm pushing the wrong button, sorry for that. You can see here on the top that ILC is E-cadherin negative, whereas this duct is positive. And you can see this is LCIS. The tumor cells are negative, and the cells surrounding it, which are the myoepithelial cells, are positive. So this is the first seminal finding. So the second one was the finding of mutations in this disease. So the first one was reported by Kanai already in 1993, which you can see here. This was only a somatic mutation, so they didn't know at that time whether it was functional. But the real finding came from Geert Berks in the group of Frans van Rooij here in Belgium. They made the actual connection between the existence of somatic mutations and the lobular phenotype. And then in 1979, Carla Vos in the Department of Pathology in Leiden was the first to make the finding that LCIS already contains these inactivating mutations. And this is important because it tells you that already at this stage, E-cadherin is lost, and it's probably causal to the development of the disease. And then 10 years later, during a previous life, In the Jonkers lab at the MKI, I provided formal causal evidence, which Christine already alluded to, by knocking out E-cadherin in a conditional mouse model with p53 in the background, and this induced a beautiful lobular phenotype with all the ring bells and whistles that accompanies lobular breast cancer. So, long story short, it ends up being like this, where ductal breast cancer is usually E-cadherin positive, and lobular breast cancer is usually E-cadherin negative. And we, of course, know this, all of us, and we've developed a white paper last year from the working group, a pathology working group from the ELBCC, where we thought, okay, we have to pinpoint what is the best diagnosis for ILC, because it was still out there with opinionated people using the wrong antibodies. So what we did is use the 5 most prominent antibodies, mapped them, and then found out which ones were the most specific, and out of the 5 we used, this ECH6 and NC38 were the best. And based on this, we provided a standard operating procedure, which we published in Modern Pathology last year, and you can also find it on our website, where we have a very simple designated flowchart on how to differentially diagnose lobular breast cancer from a histopathology point of view. So we want to push this through the channels of the World Health Organization to make this the standard in the field. Okay, so I think it's really, especially within this audience, it's good to have some historical awareness of historic findings. It's just good to know your history, that's one. And I always do this with undergraduates as well, to teach them how to cite proper papers. So if you are telling people about something about E-cadherin or lobular breast cancer, cite the seminal papers from the past and not a review from 2022. At least that's what I propose. So just to put you back into history a little bit, so the first finding that adhesion was actually important in cell-cell contact was done by François Jacob. He's a Nobel Prize winner for something else. But they had a very incidental finding in the lab that I think probably a grad student forgot to put calcium in a medium or something like that. And came back the next morning and saw that these cells were completely dissociated, like this. So this was a finding that back in the day you could publish in Cell, which was really nice, and it's a real seminal finding in the field. And then the next finding was by the group of Walter Birchmeier in Germany. They had an antibody called anti-ARC1, and they mapped it to a protein called euvemorulin, which later turned out to be E-cadherin. If they put the antibody onto cells that were adhering, the cells would dissociate. So that was the first indication that there was something keeping the cells together, and when you meddled with it through an antibody, you would impinge on cell-cell adhesion. Then Masatoshi Takeichi in 1990 cloned the gene and then called it E-cadherin for cell adhesion molecule. I don't know where the IN stands for. And then Rolf Kemmler did the same thing, but he was too late publishing the actual cloning. But what he did do is combine the two. So he combined the calcium finding from François Jacob with the adhesion findings that Birchmeier did, showing that if you mutate the calcium site in this euvemorulin being E-cadherin, the whole system collapses. So this is formal evidence that E-cadherin is really important for cell-cell adhesion. And then the next finding was again by Walter Birchmeier, and they made the first assumption that it has to do something with invasion. So they showed that if you have low levels of E-cadherin, which are these cells, that they are very invasive in a gel. And if you have cells that are very high in E-cadherin, they're not that invasive in a gel. And if you put an oncogene in it like RAS or SARC, then they will lose E-cadherin and start to invade. But this was still an observational observation in that sense, so there wasn't any formal evidence. So the next piece of evidence came from, again, the group of Frans van Rooij in Ghent. I think he's retired now. But anyway, Chris Flemings in his lab made the first actual formal proof that if you do a genetic inhibition of E-cadherin, you get invasion of cells. So what you see here is a very old-fashioned experiment. It's called a chick heart invasion assay, and what you then do, inject cells into the chick heart and see whether they invade. It doesn't invade here, but if you then meddle with E-cadherin expression, you see that it starts invading. So this was the first formal proof of that. Okay, so a bit on what E-cadherin does. E-cadherin keeps cells together. That's what it does in a normal situation and in some types of cancer as well. So what you see here is two cells. In green, you see the cytoskeleton of the cell. In blue, you see the nuclei of the cells. And in yellow, there's E-cadherin. E-cadherin is placed in between the cells. And this is the first point of contact, what you see. And you see the cells start pushing, and that's why you see these wires. So if you do a cartoon close-up of this, you can see that an E-cadherin molecule binds to another E-cadherin molecule on another cell, and then it binds that to the actin cytoskeleton. So it provides the linkage between the cell skeleton and the other cells. That's basically what E-cadherin does. Now you can see, and I told you already that this is calcium dependent. It's dependent on a very specific site in this region. And we modeled this in our lab, and what you see here in real time is the dynamic interactions between one alpha E-cadherin molecule in blue and the other E-cadherin molecule in red. And what you see here in orange are the calcium ions that are needed for the actual interaction for the X dimer. And this is what actually happens in real time. So if you put this down in a cartoon again, it looks like this. And the reason I'm putting an extra cartoon in here is because I have to introduce the catenins. The catenins are molecules that are absolutely essential for the whole function of this complex. First, you have alpha-catenin. You have alpha-catenin that binds E-cadherin to the cytoskeleton, beta-catenin, and delta-catenin, which we also call P120, and I will come back to that in a minute. Take-home message is these catenins are absolutely essential for the stability of the whole complex. Okay, so what happens if a cell encounters another cell and it undergoes an E-cadherin interaction? So the first thing that happens, it touches the other cell with the E-cadherin molecule. And then it starts pulling and pushing. So you get an increase of forces, which then opens up alpha-catenin. It sort of stretches out and then allows vinculin, which is another molecule, to come in. And vinculin is a mechanosensor. So the more tension there is on a complex, the more vinculin will come in. So that's the first thing that happens. And you see that the forces are building up. So the cytoskeleton is pushing and pulling on this molecule. So the next step that happens is that you get recruitment of a factor called ephedrin. Ephedrin binds to alpha-catenin and basically forms a stabilization complex with this alpha-catenin E-cadherin molecule, and then allows vinculin to go off again. So vinculin comes in, tension goes up, vinculin goes out, and the whole system starts relaxing, and then you end up with a stable junction like this. I'm cutting corners here scientifically, but this is the gist of the whole E-cadherin interaction. Now, what about E-cadherin? So E-cadherin is mutated in 60 to 80% of all lobular breast cancers, and we have formal evidence that this causes ILC. So what about the others? So first, P120 catenin, because it's a very prominent member. P120 catenin is absolutely essential for the stability of a classical cadherin. That can be P-cadherin, N-cadherin, OB-cadherin, M-cadherin, all of the classical ones. So we knocked it out together with L. Reynolds. So L. Reynolds did a knockout mouse, we did the breast cancer modeling, and what we saw is that we developed a very high-grade multinuclear metaplastic carcinoma, so we never encountered any ILCs. So P120 is not important for ILC in that sense. To the best of my knowledge, no one has ever tried to knock out beta-catenin. Someone tried it in a polyoma middle T model, but it never developed into breast cancer, which was lobular. And the other thing is that we hardly ever find inactivating or activating mutations in beta-catenin in lobular breast cancer. These two guys are quite important, I think. So it's been now well established that alpha-catenin is very important in this process, not only in lobular breast cancer, so we could show that if you knock out alpha-catenin, you get a lobular-like phenotype, but it's also a germline mutation in hereditary gastric cancer. So it seems that alpha-catenin is one of the key players. We now have the same evidence for ephedrin, a paper that will be published hopefully soon, which I talked about last year. Same thing, if you remove ephedrin from the equation, the whole system is disbalanced and you end up with a lobular type phenotype. And I put a little asterisk there because ILC that is dependent on alpha-catenin and ephedrin ends up to be E-cadherin positive. So these are the 10%, I think, or a part of the 10% that are E-cadherin positive ILCs. And the final one is vinculin. That hasn't been done. Well, it has been done in my lab, but we haven't published it. If you knock out vinculin, you also get lobular-like phenotypes. And we found mutations in vinculin, but they're somatic, and we're not quite sure yet whether they're actually a dominant active or dominant negative. So maybe more on that later in the coming years. Now, all of this is super nice, and we know that this works, but there's a big if and but. So E-cadherin loss is not tolerated in the mammary gland. If you lose E-cadherin, if you model it in a mouse, for instance, all the cells that target E-cadherin are apoptotic. So it's not interchangeable with life. So anybody in the audience know why that is? And maybe the more important question, what should happen before you get cancer? Does anyone have an idea? Scientists can answer the question as well. Ron.
You need a survival factor, a metabolic factor, to allow the loss of E-cadherin to be tolerated.
Yes, exactly. So if it's not tolerated, you need something that provides the tolerability. So you need something that basically tells the cell it's okay to lose E-cadherin and you can still survive. So and that boils down to this scheme. Again, it's a bit oversimplified, but I think the take-home message is very valid. First, you need a genetic inactivation or an activation of an oncogenic pathway. In breast cancer, and particularly in lobular, it's activation of PI3 kinase, amplification or mutation of AKT, a deletion of PTEN, or whatnot. I think that's about 60 to 70% of all lobular breast cancers. The other 40 are comprised in part by loss of the P53 activity, either through deletion, mutation, or amplification of MDM2. So if that's the baseline setting, then you're allowed to lose E-cadherin. And that's what happens, I think, in real life. So the main player is E-cadherin. That's 60 to 80%, and then we've been chipping away at these other markers. So catenin alpha-1, so alpha-catenin, afatin, vinculin, and the other one is PAR3, for which there is also a little bit of evidence that it induces lobular type invasion. So then you're almost a lobular breast cancer. So we know in the mouse 2 is almost sufficient, maybe a third hit is required, but we know that in humans you need about 5 to 8. So on top of that, you will acquire during the years mutations in other genes, like TBX. I'm a bit of a donkey here, aren't I? So TBX, FOXA, GATA3, and all the others, which are renowned and known tumor genes or tumor suppressor genes, but it is still unclear in what's the effect on the actual cell being lobular and the lobular phenotype. And then, of course, the treatment starts. And treatment is a renowned inducer of additional mutations, such as things we've been talking about yesterday. So estrogen receptor 1, I think ERB2/3 is a nice progression factor. Also, we see more mutations in P53 and IGFR1. So it's the first hit, inactivation of the junction, and all the things that basically propel the progression of the disease. Okay, so it's not a one-trick pony. I just want to emphasize that it does a lot of things. So the phenotypes you already know. If you have E-cadherin, usually you get more circumscribed tumors. They are invasive, but locally invasive, at least in the experimental setting. And the reason for that is that if you put them on a plate, they grow like a cobblestone epithelial formation. They stick together. If you put them in a suspension condition, which you see here, most of the cells die. And that's the reason why they don't metastasize in a mouse like this. If you look at the E-cadherin models, it's the other way around. So E-cadherin is lost, you get a very nice lobular phenotype, you get metastases. If you put these cells on very stringent substrates, they are very, very motile, which you see here. And if you put them in suspension, they're okay. So they're really rigid. One cell can survive in suspension for weeks at a time in the lab. You can come back, forget about them, put them back on a plastic plate, and they will grow again. That's typically lobular for you. Now, underlying this is a lot of things. So first of all, we know that mRNA transcriptional profiling is different in E-cadherin negative cells. So that's one thing. We also know that it impinges on protein localization. So an example here is P120. With E-cadherin, it's nicely on the membrane. If you lose E-cadherin, it's translocated to the cytosol and the nucleus, which is a typical ILC thing. We also know that cell cycle progression is affected. So this would be a typical IDC with a lot of phosphorylated RB in the nucleus, whereas lobular breast cancer also sometimes have cells that divide and have phosphorylated RB, but most of the cells have cytosolic RB, although it's phosphorylated, which is really weird. Lastly, something that is hip and trending in the last years is the extracellular matrix. There are clear indications that ductal have a different composition of ECM molecules, which you can see here in green, which are the collagens, the laminins, the fibrillar fibronectins, and so forth. And whereas in ductal, it mostly looks like this, in lobular, you see more of these different patterns. So there's clear differences underlying all these phenotypes. Okay, So diving into the molecular mechanism, and this is important for the things I'm going to talk about in the preclinical setting and in the clinical setting. So first of all, I have to talk about ER. It's not my forte because I don't think ER is directly linked to E-cadherin, and I could fill a complete seminar with ER, but I just want to emphasize that it's the most important thing clinically, right? I mean, the disease is manageable because ER vulnerability is there. There is some specificity between IDC and ILC. I think there's beautiful work from Rina's group, Rina Jeselson and Matt Sikora, showing that there might be specificity for a factor called FOXA1 inducing heterochromatin condensation and super enhancers, which might be specific for ILC. But I'm not going to go into that today. I'm going to stick to what I know, and that is E-cadherin. So what we know is that if E-cadherin is lost, P120 goes to the cytosol. I just briefly showed you that. And the first thing it does, it induces actin contraction. So basically the cytoskeleton starts pushing and pulling, although there's no upside Q, right? So there's nothing here, but still it starts pushing. And it's very typical for lobular breast cancer cells. We know the molecular mechanism. It's been published a long time ago in 2013. I'm not going to go into that. But the bottom line is that Rho kinase is activated as a central player. So the second thing that happens because of this contraction, you get nuclear localization of a factor called YAP. And YAP is a hippo signaling targeting factor which goes to the nucleus and then basically induces transcription of all kinds of genes. And it's sort of a yin and yang of contraction. If you have high contraction, YAP goes to the nucleus. If YAP is in the nucleus, you will get high contraction. So it's a self-propelling mechanism. And why this is important, I think, is because if you look in normal breast cancer cells, you can see that luminal cells never have nuclear YAP. YAP is in red. Whereas the myoepithelial cells, the ones that are pushing out the milk and have a lot of contraction, they are YAP positive. So a luminal cell shouldn't have YAP in its nucleus, and lobular cells do. So that might be a cue for future interventions as well. We know also that P120 goes to the nucleus where it induces a very broad transcriptional program of differentiation genes that inhibit proliferation. And we published something last year on maybe a rationale why ILC is so low proliferative. So what we found is that a factor called ID2, which is induced upon P120 Kaiser interaction, directly interacts with RB and prevents phosphorylation through CDK4/6. And we think that's the reason why ILC is so slow proliferating in certain settings. Another thing that is really important, but not a lot of work has gone into it already, are the integrins and the ECMs, so the extracellular matrix. So it's not just E-cadherin on the outside, it's also an integrin on the outside. And it basically attaches itself to its environment. So ILC might have specific ECMs, it might have specific integrins, well, it does, but it's not being published that well. And we know that it activates factors like SARC and FAC, which are again upstream of Rho kinase. So this might be a feed-forward loop in the sense that if you lose E-cadherin, things start contracting, you have specific integrins, and once they get to a certain location, they will propel this pro-survival phenotype. And finally, something I want to go into a little bit further today is growth factor receptor signaling. I think growth factor receptor signaling is super important in ILC. Just to show you a brief overview for the people who are not connoisseurs in the group, you have a growth factor receptor that binds to a growth factor. It signals to downstream effectors. One of them is PI3 kinase, which you all know, I think. And then it acts upon a factor called AKT, or used to be called protein kinase B. And this propels, again, phosphorylation and dephosphorylation of all kinds of proteins that are important to the cell. And the bottom line is that it induces activation of either apoptosis if it's inactive or survival if it's active. Now the next slide has nothing to do with lobular per se, but I just want to illustrate how complex and dynamic this is, because you can already see that the feed forward and the feedback loops in here are quite extensive. So this is a couple of movies from my buddy Olivier Pertz in Bern, and what he's been doing is magnificent work on activation of growth factor receptors in real time. So what you see here is a solid tumor, a breast tumor. This is the displacement of the nuclei, so it's just wiggling back and forth. But it's not random, it's completely orchestrated. So one thing you can see here, that the pulses of AKT and the pulses of ERK, which is another downstream target of these growth factor receptors, goes in waves. And it's very dynamic, and it's very orchestrated. And once you put a factor in it like alpeculib, which is a PI3 kinase inhibitor, you can see that the whole thing collapses, but only partially, and the waves are still propagated. And this might be one of the reasons why these drugs are not performing up to standard in the clinic, because there's no real clear benefit in the sense that if you are mutant versus if you are wild type. So these, just to realize that this is super dynamic and complex to look at. It's not on and off and we put a drug and it's gone. It's not like that, okay? So again, this pathway. So what can you then inhibit from a clinical point of view? So the first thing people look at is the proximal thing, and the proximal thing being the growth factor receptor, which is on top. So the first thing that was done was this landmark paper by Chris Lord's group. So magnificent work looking at synthetic lethality, which basically means which factors are important to propel your phenotype in an E-cadherin negative background. So they started knocking out things, and one thing they found was ROS1, which is an orphan receptor tyrosine kinase. And of course, there's a drug against everything, also against ROS. It's not very specific against ROS, but it inhibits ROS, and that is crizotinib. And they found this in the screen as well. So beautiful work showing that there's a vulnerability that can be targeted if cells are E-cadherin negative. And from this, 2 trials were devised. The first one is ROLO, which has been running for quite some time now, using crizotinib at the ICR, and the other one is Rosalind at the Institut Jules Bordet using entrectinib, which is also a ROS inhibitor. I won't go into the details because A, I don't know them, B, I don't know whether I can share them with you, so go harass either Chris and Philippe about these trials if you want some more information. Okay, so that's one thing. The other important factor is IGF. So both my lab and Adrian Lee's lab have found that if you look at this disease, the one factor that really pops out is IGF. So if you have low E-cadherin, so this is TCGA, but you see the same thing in Metabric and the others, low E-cadherin means high IGF. The receptor levels are about the same, but Adrian showed very nicely that this coincides with activation of the receptor, which you see here in red. So lobular breast cancer has high levels of IGF and higher activity of IGF-1 receptor. So you would think, okay, cool, this is it. I mean, this is clearly very important. There are inhibitors against IGF-1R. Why not use this clinically? Well, there's one big caveat, and that's these things are quite toxic from a metabolic point of view. So if you use antagonizing molecules against IGFR signaling, you meddle with insulin signaling as well, and this messes up your whole metabolic pathway. So this is not the way to go, but I think the promising thing is that IGF is still very potent, and we might want to revisit the monoclonal antibodies against IGF or think out of the box to inhibit IGF signaling in this disease. Okay, so coming back to other things that could be inhibited in lobular breast cancer, I've briefly talked about PI3 kinase. I personally think it's not the way to go. We can talk about this during coffee, but I do think that AKT is very important. And I think it's important because it's very central in this feedback and feed-forward looping activation. So I'm showing you this ridiculous slide because, yeah, it's a ridiculous slide because it shows you everything that AKT activates or is inhibited by AKT or something that inhibits AKT and so forth. The reason I like this slide is because AKT is at the center of the universe, and that's what I wanted to talk about. So, this is a bit about AKT. AKT is a molecule that looks like this. It has a domain that basically does a linkage to the membrane, which is not that important for now. What is important is it has 2 phosphorylation sites. The first one is the threonine, which is activated by PDK1. The other is the threonine, which is absolutely essential for full activation and is activated by mTOR2. And you can already see where the problems start arising, because mTOR2 was identified as a downstream target for AKT, but it's an upstream activator of AKT as well. So it goes back and forth, and that's really challenging. Okay, the good thing is we have a lot of inhibitors. They've been talked about already during this meeting yesterday. The first one I put down here is MK2206, which I think is absolutely the best AKT inhibitor, but it's also super toxic. And that's because it also inhibits mTOR, which is not very good for patients. The ones I'm going to be talking about today briefly are these ones in green, which is, maybe Claire should pronounce these. Where is she? She loves pronouncing these names, no? Okay, so this is called capivasertib, which is AZD5363, and the other one is ipatasertib. And they are pan-AKT inhibitors. So they inhibit AKT1 and 2, which are more metabolic and survival pathways, and AKT3, which is specific for the brain. So I'm going to talk about these as clinical intervention tools. So we know AKT is really important, because AKT was already identified by Chuck Peru as a very central player. My lab and Adrian's lab showed the same thing, but then showed causality. So if you knock out E-cadherin, you see that if you put IGF in the system, you get a massive upregulation of activation. And the experts tell me that you can multiply these signals, so it's about a 30-fold induction. These are mouse cells. We see the same thing in humans. Adrian saw the same thing in humans as well. It's a little bit less because, for instance, this one has a PI3 kinase activating mutation, and then you see a little less activation as if you would if the system were wild type. The inhibitors work really well as well. So in the knockout situation, you have a 10 to 20-fold reduction in increase in sensitivity, so you can use less drugs. And again, this is causal. If you knock it out, you see the same thing. And we did the preclinical mouse experiments in my lab. There's this one in Adrian's lab, and we see the same thing. So they're very effective in inhibiting growth in lobular breast cancer. Okay, so we thought this is really nice. We can do a trial, and I see time is up, but I'm not finished yet. Sabine Lin at the NKI was the lead investigator in this. We wanted to do a trial, AKT and ILC specific using capivasertib. But unfortunately, some of you know the traumatizing experience we had in the group. When this happened, 2 months before the start of the trial, this paper was Prof paper was presented at the San Antonio meeting showing the Capitello trial. So basically what they did is exactly the same, but they did it in the whole ER positive, HER2 negative group. And they saw really nice results. So basically, the necessity for Pfizer to do a trial on lobular was completely gone, which is, of course, really saddening. What is more saddening is they either don't read literature, or they don't read my literature, or both. But they had an inclusion criterion that the patients had to be PI3 kinase mutant, or AKT mutant, or P10 mutant, which is exactly the group that will not benefit the most from this treatment, based on the biochemistry we did. The interesting thing is, if you look at the 13-page paper and the 200-page protocol, there's no mention of lobular whatsoever. So I had a talk about this with Mafalda, who's the first author on the paper. She either doesn't want to tell me or she doesn't know, but she told me that there were a surprising number of E-cadherin mutant cases in the responder group. And they will probably publish it somewhere this year or next year. So I'm putting my money on the lobulars. So finally, I'm sorry I'm going over time a bit, but it's just 2 slides. Can we do something with this laziness? So we know there's a very strong dependency on RB phosphorylation in lobular breast cancer, experimentally at least, and in clinical samples. So can we do something for targeting? So there's already some indications that there's a very counterintuitive response to these drugs. So these were 2 papers published in 2009 and 2018, Palbo and abemaciclib, which are 2 CDK4/6 inhibitors. And surprisingly, all the E-cadherin mutant ones are on the left. Apart from SKBR3, which is a HER2 amplified one, the best responders are the ones that proliferate the least, which is, to me, was mind-boggling. So, okay, let's find out if there's an actual causal link between the 2. And we think there is. It's unfortunately still unpublished, but I would like to show with you some data that E-cadherin loss, we think, causes palbociclib vulnerability. So this is a lobular breast cancer that is E-cadherin positive. This is a lobular breast cancer that is E-cadherin negative, and you can see the E-cadherin negative one is way more responsive to the drug. And this is causal evidence. If we knock out E-cadherin in the E-cadherin positive lobular, it gets more responsive to CDK4/6. So we think this is super exciting. And why is this super exciting? Because we can think about combinations. So this is what I think lobular is all about. It's a slow-growing disease. It goes into the bloodstream. It's completely dormant. It doesn't proliferate anymore. It doesn't die. It ends up somewhere where we don't understand what happens. And then somewhere along the line, it starts dividing again. So it's always vulnerable for AKT. It's very vulnerable for CDK4/6. So can we combine the 2 to see whether they are very effective? So this is experimental evidence for this. These are very complicated isobolograms. I think the take-home message here, if you see blue, it's synergizing. And what you see is that the bluishness is most prevalent in the ones that are E-cadherin negative, meaning that palbociclib together with capivasertib is synergistic if you knock out E-cadherin, i.e. lobular breast cancer. And final thing is, we did this also in mice. If you do both drugs, you see this, very nice inhibition. It's not better than ductal, but the fact that it's very effective at all is surprising, I would say. And then we did the actual experiment by looking at relapse, and then we saw a very spectacular thing. So what you see here is the ductal ones. If you wait a while with treatment and then start treating again once they get 300 millimeters, they all progress to rapid disease, whereas the lobular ones remain as a stable disease. It's very toxic, mind you, so we have to still find out what's the best regimen and so forth. Whether you can do this at the same time is also clinically very doubtful, but I think from a proof of concept it looks really promising. So I end with this. ECAT hearing loss is key. It's the thing that's driving the disease, and it's only one of the diseases where we actually know this, so we have to take that into account when developing things. We need a better understanding of the driving mutations along the line. Are they drivers, passengers? Are they invoking chemotherapy resistance, estrogen resistance, and so forth? And of course, an open door is to diagnose and treat according to biology and not just looking at things. So looking at survival pathways, I think, is absolutely key. The use of BH3 mimetics has been completely neglected in the lobular field as well, I think. How is dormancy regulated? What are the survival signals? Is there an extracellular matrix component? What are the immune cells doing in this entire party? I mean, it goes on and on. We need more money, we need more research. And there's the long latency relapses, which is the actual clinical problem. How does that work? Can we target the cell cycle vulnerabilities, chemoresistance and inflammation, and so forth? Okay, final point is that inclusion criteria. This has been bugging me as a biologist for a long time. I mean, if you know that PI3 kinase mutation inhibits your responses, why do you exclude these people from your clinical trials? It's the same with triple negative ILC. Triple negative ILC is not triple negative breast cancer. It's triple negative ILC. So if it has an ILC vulnerability, treat it as such, and not just as a triple negative cancer. At least that's my opinion, but you can take that away however you want. Okay, so final slide. The people in my lab, I want to thank. We have a vacancy for a PhD student, so if you know someone who wants to do a very cool PhD on lobular breast cancer, please come and see me. The collaborators, some of whom are in the room, and the sponsors. And with this, I'm over time. Sorry, Christine, but I'll take your questions. If there is time for questions.
No, we will actually not do questions because but I thank you very much and I congratulate you again for this lecture, which was, I think, really a tour de force and a very nice story actually of everything he has accumulated and a look forward for what we still need to do. So now I would like to invite actually the chairs for the next session. Dr. Philippe Aftimos, Dr. Jason Muabi and Leigh Pate, so that they could introduce the next speakers. Thank you.